Welcome to the lab. I'm Drew Cullip. Today we have a special opportunity. Our labs have partnered with an ostrich farm to produce some applied research in both cosmetic science and in antibody research. As a result, we had some ostrich eggs in the lab recently. Today we're going to open one up, see what it looks like on the inside. These are not fertilized eggs, but I thought it was going to be interesting anyways. I have a chicken egg beside it in comparison, so hopefully you can tell which one is the ostrich egg and which one is the chicken egg. Let's open it up and see what it looks like. First off, I want to open up the chicken egg. I want to do a comparison between the volume inside a chicken egg versus an ostrich egg. We'll open this the same way we're going to open the egg for our primary culture. I don't care about sterility here. All I want to know is, what is the volume inside a chicken egg? Crack it with a pair of scissors and then peel off the shell with some curved forceps. Then I'll dump the egg out in the beaker right beside it and we'll measure the volume. Peel the chorionic membrane. Notice how thin it is. Take note of that. The ostrich egg one is much thicker. It's like tissue paper. Very, very thin. Now that we have it open, we'll invert it and dump the contents into this 100 ml beaker. As you can see, it's just a standard non-fertilized egg. And the volume, just about 50 mils. Just a little bit less than 50 mils. Maybe 50 mils right there. So we'll say the volume of a chicken egg is 50 milliliters. Let's now see what we get when we open up the ostrich egg. Also, let's see how we're going to open the ostrich egg. It's going to be very different. As you can see, we have to use a much larger beaker to hold that in place. We're going to candle the egg to start with. If you saw a previous video, you saw that the chicken egg has an air pocket on the rounded part. Let's see if the ostrich egg also has something very similar to it. I can tell you this, it was extremely heavy. It's much heavier than you think. We have to turn off the lights to candle the egg. And here we have our egg candler. Again, it's just a high power LED light. And you can see there's that air pocket with our chicken egg on the rounded part of the egg. Let's see what happens when we candle an ostrich egg. There it is. It also has an air pocket. I'll be honest, both ends were rounded on this one. I could not tell a pointy end like what we have with the chicken egg. So to find out where the air pocket was, you have to candle both sides. We're going to open this the same way. We're going to crack it open where that air pocket was. Again, when we're hitting the egg, it will potentially damage it. We don't want the liquid to spill out everywhere, so that's why we have that air pocket region where we can crack into it. Now, we have something different here. We looked online on how to open up an ostrich egg. They were using power tools, and we got a meat cleaver we saw online, and that was an option. So we have some students here who are going to crack it open, hitting it with the blunt side of a meat cleaver. We weren't sure how hard to hit it. The answer is you got to hit it very hard. Look how hard she's hitting it. That's a very tough shell. One thing you can see as well, as it's being cracked open, is you can see the membrane inside. And I can tell you this, the membrane is much thicker in the ostrich egg compared to the chicken egg. I will say none of us had experience opening up an ostrich egg at this point, so everything here was just a learning experience. We had a large audience as well in the lab, so you can see some of them in the background. You can start to see how thick the egg shell is. We're going to peel off with curved forceps. And look how thick it is. We'll peel away the shell where that air pocket was to reveal the chorionic membrane. And unlike the chicken embryo, where the membrane was very thin, this membrane is very thick. In fact, it kind of felt like a vinyl balloon. I tried to instruct the students to open it large enough so we could pour the contents out. Here we go, we're going to rip the membrane. You can see it's much thicker. And as we peel it, you can see there is our yolk. We weren't sure what size of beaker to get. Someone suggested a 1 liter. I thought a 2 liter might be better. Let's see what the volume is. Look at that. That's a lot of egg. Let's see the volume. Almost 1.2 liters. That's 2 dozen chicken eggs compared to what we had from before. You can see inside. And it looks very similar to the chicken egg. Just everything a lot thicker. The yolk appeared to come out, staying separate from the egg whites. It does look a bit different, but it had to be squeezed out through that small hole. So it was kind of stretched out as a tube, I think. That's why it looks this way. Now, for the applied research, they did need to separate the yolk from the egg white. That would make a big omelet. There's the membrane. Again, it was almost like a latex balloon. That's what it felt like. Very thick and rubbery. You can compare the size of the ostrich egg here to the chicken egg. And as I was doing this, I was thinking perhaps this is what dinosaur eggs were like in size. Here we are trying to separate the egg whites by just pouring it off. We had some other options, but there was just too much volume perhaps to use some of the other options we were planning. So we thought just to pour it directly off. Worked out pretty well. Here's some of the egg yolk. They're trying to mix that up. And here's the egg white. Quite a bit there. Not something you see every day in the lab, I'll tell you that. Another goal in this lab was to try and remove any of the fibrous tissue from the egg white. There was a strainer brought in. Oh, here we go, just to see if the... I thought that was funny, the egg can fit directly in the hole. Anyways, a strainer was brought in, and we thought we could try and pour the egg white through the strainer. It started to work. Let's get a little closer there. Just pour it directly through. And you can see the egg white did come through. However, it got pretty clogged up. After the camera goes off, I actually rigged up a 1 liter sterile filtration unit, and I stuck this in there, and we were able to use vacuum pressure to pull the egg white through there, trying to remove some of the residual fibrous tissue that was left over in the egg white. That's one of the fun parts of working in a lab, is working on the fly and making things up as you go. Later on, we actually peeled the chorionic membrane directly out of the egg, and here it is right here. We're going to dry it out, place it over top of an Erlenmeyer flask for it to dry. You can see our sink is quite messy, so I made a joke and we moved it over to the other side. But it was able to come out as one piece. We just peeled this out directly. It was able to separate from the eggshell. It was an amazing day in the lab. And again, it was like a white latex balloon. That's what it felt like. I almost break the egg here. Here it is. You can see we've removed the membrane from inside the egg. Just eggshell. And I don't believe any of this will be wasted. All the products here will be used either in the cosmetic science projects or in the antibody research. So I hope you enjoyed this video. It was a unique day in the lab, comparing both the size and the volume and the differences between a chicken egg and an ostrich egg. Until next time.